Ansgar basically, one second, got it. Yeah, Ansgar taught me a very important life lesson. And you know, like we're all driven by the desire to make discoveries and we enjoy doing science, etc. But really, as you get older, like you realize that the real magic of science is to make good friends and to work with like-minded people, and this continues throughout your life. In fact, you always have a team of smart people, and Ansgar is the prime example. So we were postdocs together in Edmonton, Alberta, and I remember one day we're like sitting at 8 p.m. on our desk and discussing how could removal of the hindwing tegula change the flight network, and we had suddenly this idea that probably the forewing makes more connections, and instead of going home, what we did, we went to our rigs, and Ansgar within 10 minutes got his 515. I got my 566, and then we stained them, and then at midnight we looked at our Lucifer fill, and our friends 515 and 566 had much bigger dendrites, and so basically by midnight we had a paper already like conceived, and then it was just getting ends. And throughout this process also I learned, working with Ansgar, that the team is the more important thing than the discovery. And there was one more example I want to give you, that when you do intercellular recording, the best thing is you pop into that cell, and then your membrane potential goes to -70, and there's no better feeling than that drop to -70. And so I remember that moment when Ansgar like had his first recording, like coming from Bessler's lab, and it popped -70. He gets extremely excited and calls Nino, where the hell are you? And so he runs through the hallway, like I was like copying somewhere in the end of the hallway, and then it took him like 20 minutes to get me. And at the moment I entered the room, he lost the cell, and I remember that, this team playing was more important than that recording at that given time. And I'm not surprised that Ansgar was extremely successful like throughout his career, because I know that he's not that kind of like hierarchical German professor that dictates around, but instead really is a team player. And if I would start all over again, I would probably come to Ansgar as a postdoc. So Ansgar, just to his CV, he did his master's in Bielefeld with Hol Kruse doing cybernetics on insects, did a PhD with Ulrich Bessler, and then he came to Edmonton, where we had incredible fun working on the flight system, and then he went back to Kaiserslautern to work on the stick insect and became finally a professor at University of Cologne, where he's been ever since, I think since '98. Amazing. A lot of consistency. So Ansgar, it's a great pleasure. I can't wait to see your talk and what you can tell us. Okay.
Good. I think I will have to do it again. This always happens. No, wait a minute, wait a second. Here we go. But you can at least hear me, can't you?
That's not enough, Ansgar. Now we can see it. Good.
Yeah, but it's not finished yet. Here we go, and then we.
No, it's not it.
Now you have it.
Yes.
Yeah, perfect.
Yeah, okay, good.
Amazing. Good job.
Amazing. I made it. Yeah, I played a bit too much. Anyway, so thanks a lot for inviting me, and it's a big pleasure to be with you. And yes, so Nino and me go really back, as John Simmers would say, 400 years and together. And what I would like to do with you tonight is, as many of us who gave lectures do, give a bit of the background where I come from, and sort of in sort of who influenced me, who were collaborators that made quite an impression or impact on me, and then tell you 3 stories of the work that we did, let's say, in the past 10 years. And so let's get started. And actually, and this Eve may recall, because I was brought up and educated in times of firm dogmas. And this is now the 80s, about the role of dedicated circuits, sensory feedback, and CPG. So I did, as Nino said, my diploma thesis with Hall Kruse, who did beautiful work on the concept that rhythmic locomotor behavior can be sufficiently explained by the role of sensory feedback, as modelly studies show. As you will see, there's no CPG here, okay? And then I did my doctoral thesis with Bessler, who again was right inside this scene of dogmas, with his finding that there's state-dependent flexibility of simple reflexes, okay? And that these simple reflexes can essentially be modified in a state-dependent fashion, which questions the concept of dedicated sensory motor networks. And now comes Eve into play, because the year I met Eve is 1989 in Tutzing. And Eve, you may for sure recall the heated controversies between colleagues bound to such dogmas at this Tutzing meeting. You recall it, Eve? The meeting after the ISN meeting in Berlin, when a couple of sort of central and peripheral people were really fighting each other, and the dedicated and distributed people. So it was fun, but for a young
Eve
guy,
was very upset that she couldn't get a salad. I remember that in Germany.
You guys remember everything, and I don't remember anything.
Oh, I know,
Oh.
desperate. No salad
Oh.
for you, Eve.
Eve, you were so cool, and the old guys, and well, it was at that time mostly guys. They were so agitated and they were so severe. Anyway, so after that, I went for postdocing with Kia, and because actually that was a cool, for me, a cool move, because the Locus Flight Network actually offered everything. Sensory feedback interaction with the CPG that enables the generation of the functional motor output for wing beat. And yes, this is when my story with Nino began. I heard from him before when he was a grad student with Jenny Keane in Regensburg, but then I finally met him. And since then, we've collaborated for some time and have been friends ever since. And as Nino already told you, I worked on regeneration of the locust flight following partial de-efferentation. But there are more and other influencers and collaborators that came later. And this is a picture taken from John Zimmer's 70th symposium. Mikey, Abdel, John were very important to talk to, and their publication had quite an impact, as had the work and later on the friendship of Jochen Flüger, who unfortunately died at the beginning of this year, briefly after a heart attack. And actually in the past 5 years, or 10 years, we got closer and closer friends. And actually this is Jochen cooking dinner in our holiday apartment where we stayed for the party at John Zimmer's house. More on the sort of science side, important impact had people like Stan Grillner, where I actually started to lamprey spinal cord work. And then Urian Eckeberger and Sylvia Down were those in the late 90s, early 2000s that introduced me to modeling. And that was actually a cool time I had for a year with Urian in Berlin, where I learned even to program Python, which I probably most of it have forgotten by now. And then Scott came into the scene by making it clear to me that, yes, Ansgar, action potentials of neurons matter, but don't you think the muscle that they drive is also relevant? And since about 2004, we have been steadily collaborating, and it has a miraculous cooperation and friendship as well. As has been a bit later when Sasha came into the scene, and still then he's trying to turn me into a sensory physiologist, but I still want to know the connection of sensory neurons to motor neurons. So he doesn't get me in the end. And then finally, a person to mention is Barry Dixon, who almost out of the blue contacted me about 8 years ago when he had a grad student, Salil Bidaye, who found an interesting neuron that you will hear more about later on that makes a fruit fly walk backwards all day. And so he said, Ansgar, you know a lot about locomotion in insects. Would you be willing to be the external opponent of his thesis? And since then, we have been collaborating, had a project at Janelia, and great fun, and you will see some of our results. Anyway, so there's one incident when very early on, about 2 decades ago, my relation to the STG family, as it was called, was markedly deepened. And this was in 2003. Eve, now you are again becoming important because you have been the postdoc supervisor of the chair of the session that I was invited to at the East Coast NerveNet meeting. I was invited by Ralph DiCaprio, and there was a talk by a stick insect researcher from abroad to be introduced, and one phrase of the introductory words were, studying walking in stick insects is like studying swimming in bricks. And Eve, do you know who it was? There's only one person who can be.
It's Dirk.
Yeah, sure, it's Dirk. I think he's not here tonight. Anyway, tell him that I'm still laughing about that one. Three things we would like to talk about tonight.
I'm here and I still think that's true.
You are, so you can. It is true. It was cool. You made another good joke at that meeting, but anyway, we are here to talk science.
It's not a joke.
It's not. We have the discussion for this. So first, I would like to take you through a rather long part on generating the motor output for leg stepping with task-specific modifications and component neurons contributing. Then we will have a very brief excursion on biomechanics, and then we have a somewhat longer ending on multimodal processing and posture control. So let's start out with generating motor activity. So it was in between the 1990s and 2010s when I was largely trying to unravel more details on the neural basis of leg stepping in the stick insects. So we could show that there's essentially the leg muscle control system has a modular organization with individual CPGs. Then we could show that in real walking or in vivo, that the motor activity of leg stepping results from tonic depolarization drive and alternating excitatory and inhibitory synaptic drive. So inputs. And finally, and this is a slide that I think Mikey told me once, this is awful. But again, it's the fact that essentially when you divide the 4 phases of a step in 2 parts of the swing and 2 parts of the stance, what we know as of today is that for straight walking, movement and load feedback contribute to coordinating the CPG and motor activity. So having said that, I end this brief introduction on the state of knowledge about 12 years ago with showing that it was the work together with Urian and Sylvia and later on Arndt that allowed me to test these findings that were coming from mostly restraint preparations by creating together with them dynamic simulations of single or multi-legged walking simulations, and essentially showing that yes, these influences are capable of generating and coordinating stepping motor output, which does not mean that this is all. It only means that they can be sufficient. And to make this complicated scheme short and digestible, if you align those 4 phases, like starting with stance to swing transition, movement and load feedback play a big role. When it comes to swing, it's only movement feedback. When it comes to the swing to stance transition and the leg is put down, then we have movement, load, and ground contact feedback. And the stance phase is supported by movement and load feedback. So what we became interested in a while ago is actually, and which is the challenge, is that we would like to understand how generating task-specific motor activity serving behavioral flexibility is coming about. And actually, this is a question that is not answered in any creature on Earth, being it a chameleon climbing the rope or these 2 mammals chasing each other in a completely unpredictable environment. And they do a pretty good job in keeping going and keeping chasing. Okay, so we did as we usually do, a very simple we approach this problem with very simple questions. For example, like, what does it take for a stick insect to walk backwards? What do we need to change in the sensory to central control features? Or how does a stick insect cope with terrain when ground contact is lacking and when searching movements have to be done in order to find foothold? And finally, and that has been very successful, how is curve walking generated in the 6-legged stick insect? And this is what I want to talk about, this behavior here in the next couple of slides. And this work was started with Matthias Grun, a postdoc in my lab, who took advantage of a very simple and old-fashioned setup where he essentially profited from the fact that when you put a stick insect on a stick, yeah, this is not a joke, it's a stick insect on a stick, and you place it on a grease plate and you give the right optomotor stimulus, this stick insect will start to walk and it will do turns depending on the direction of movement of the stimulus. And everybody can see immediately this stick insect tries to do a left-hand curve. And you also immediately see that the leg movements are different. So there's definitely an inside kind of movement and an outside curve kind of movement. So what's the difference? When you quantify the kinematics of the leg movements here, the stance trajectories, you see that the outside mid leg does a long stance to backwards, while the inside mid leg, and particularly here in the rostral part of the plot, you see that the mid leg has a high variability in stance phase generation and does even almost backward appearing stance phases. So when you sum up what we found on doing the kinematic analysis, we could show that the mid leg generating an inside stance shows a marked tibial flexion and marginal leg retraction in many cases. While on the outside, we have a marginal tibial flexion and marked leg retraction, okay? There's another property, a characteristic becoming clear, and this is that obviously the cycle periods of the legs on both sides differ from each other. The cycle period on the inside, on the slippery surface, which is much shorter, about 50%, than the cycle period on the outside. So we did a couple of studies, two of which I will show, which essentially showed that task-dependent changes in influences of sensory feedback signals about movement and load contribute. And they are now colored brownish in this horrible scheme here. It's these brownish arrows where we could show that the influences of specific sensory feedback changes depending on the task of the leg, being it an outside leg or an inside leg. So on the left side, you see the situation for the inside leg. Again, the stance phase, marked tibial flexion and marginal leg retraction. Or here on the right side, you see the outside mid leg with marginal tibial flexion and marked leg retraction. And that's work done by Katja Hellekes, Joscha Schmitz, Philipp Rosenbaum, who later on was a postdoc for some time with Yves and Matthias. And we could show that the marked tibial flexion and the marginal tibial flexion is due to the fact that the influence of movement signals is site-specific. We also could show that the marginal leg retraction on the inside and the marked leg retraction on the outside are the consequence of the influence of load signals being site-specific. Again. So let's turn to the last point and give some evidence on the load, on the influence of load in curve walking. Here you see the animal as we had it before, except that we removed 3 legs, the hind legs, the left middle leg. The 2 front legs do walking on the slippery surface with an optomotor stimulus, so they can do a left curve or right curve. And the right middle leg stump is fixed, and we do very small stimuli which mimic load on the leg. And when you do so, and the front leg is walking, you see that when you record the promotor and the remotor activity of the middle leg, you find that always when you increase load, you will activate the remotor. Okay, this is shown here. 0 is the time of stimulation. The activity is always increasing after begin of the stimulus. And when you quantify this, you can show that in all cases, feedback signaling load increases or initiates retractor activity in the otherwise deafferented meso. So there's no additional sensory input coming in. On the inside, the situation is completely different. You do the same experiment, you just turn the stripe pattern movement, so the animal walks the right curve with the front legs, and then you stimulate the load sensors, and you find a situation which is not at all systematic. Sometimes you initiate remotor activity here and here. You can even stop it, initiate promotor activity. And when you quantify, you find a diverse picture. In 27%, an increase of remotor activity. In 54%, a decrease or termination. And in 19%, it does not affect coxal activity at all. Okay? So obviously, the influence of load signals is site-specific, with being strongly activating retractor activity on the outside and being highly variable, and thereby potentially contributing to the marginal leg retraction, or it can at least not support a long leg retraction on the inside. As well, as I've shown you before, we found another interesting difference. And this is that obviously on the inside of the curve, you have a short cycle period of the leg while you have long ones on the outside. Again, we could show that the descending drive to segmental CPGs to be site-specific. And the evidence is shown here when you have a stick insect in the same condition, but now you do not have any afferent feedback left. You have a de-afferented mesothoracic segment, and you just record the activity of promotor and remotor. You see that when you are on the inside, this is the stance phase activity of the front leg. You see that there's a very fast alternation between a promoter and remoter. While on the outside, you see there's mostly tonic activity in the remoter and as well tonic activity in the promoter. When you evaluate the correlation between remoter and promoter activity, you find that there's a negative correlation, which is antiphasic activity on the inside, but in-phase activity on the outside. So that gives rise to the idea that potentially the CPG of the protractor-retractor system could be involved. And in order to test for that, Matthias did the following experiment. He did again place the stick insect on the slippery surface, de-afferented all the caudal's ganglia. Then he generated a split bath and used pilocarpine to activate only this segmental ganglion, which generates a nice alternating activity when the animal is standing, as you will see here, as the front leg doesn't show any activity on the flexor. When he then induces an inside leg, you see that compared to the resting rhythm, you have a much faster alternation between promoter and remoter. While when you do the outside leg condition, you see that the system is almost halted or arrested in the retractor phase and only sometimes switches to protractor activity, providing evidence that in fact the influence on the cycle period you see on the inside and the outside is mediated by the contribution of the premotor rhythm generating networks of that particular leg joint. So having said that, the consequence is that at the moment we know that site specificity of changes poses the question on the role and action of descending and local mechanisms contribute. I didn't want to dwell too much into the details, but essentially what you find on the inside and on the outside does not at all mimic or is reminiscent to what you may find in straight walking. It differs in a very specific way, which means that you cannot simply have the idea that simple descending command from the brain or from rostral segments can explain the changes that you see in the influences of specific feedback signals onto the CPGs. So let us briefly talk about what we are currently doing on the control of leg stepping. Teaming up with Sasha for a large number of years, we have now gone to look into the specific load feedback signals that serve the generation of task specificity. The stick insect has 11 distinct locations of load sensors, the campaniform sensilla, and we have looked at them quite carefully together with Sasha and described their sensitivity. And what we found was, for me at least, as collaborating often with engineers, quite exciting, because the spatial distribution and sensitivity of campaniform sensilla constructs some kind of leg reference frame. This is denoted here by a scheme of Sasha, which essentially shows that when you look at the sensitivities of the campaniform sensilla, they either are sensitive within the plane or orthogonal to it. And that's interesting, which means that this nervous system essentially has to cope with a leg reference frame that has to be moved when it wants to generate a step. So what Anna Haberkorn is doing in her PhD thesis, and she's finishing as we speak, she has looked at identifying target motor neurons for all the loading directions campaniform sensilla on the leg, and looking into their specific sensitivities, for example, for promoter and remoter, which is retractor and protractor, shown by the equivalent colored arrows here. The second question that we came along was, and you may have recalled, seen it from the scheme I showed you, that not only loading information from the leg, but also unloading appears to play a role. So the question that Sasha and Nick Czecinski, together with a grad student of Sasha, have and us have answered lately is loading and unloading perceived by a campaniform sensilla? And this is shown here. The answer is shown by a recording of the tibial campaniform sensilla, which measure load on the tibia, and they were stimulated by slightly bending the tibia, and the force was measured, the force exerted on the cuticle. And you see the blue ones, the 6Bs, respond to increase in force, but the 6As respond to decrease of force. And they do so in the same way by coding for this change in force over time, dF by dt, in a way that we know today that individual campaniform sensilla report load increase, but others report load decrease, which is unloading is not a passive signal by silencing campaniform sensilla, it's an active measured sensory feedback signal. The third question we are currently going for is actually, you may say, come on, you have shown this scheme so often, you are sure that all those CPGs operate in a similar way. Well, it was more by chance that Angelina Rute and a graduate student and Charalampos Manciares, a postdoc, stumbled into looking closer into this, what we thought resolved question, because it was known since quite a while that the CPG of the motor neuron supplying the femotibia joint drives these by phasic synaptic inhibition, which is shown here by the filled circle, which means that essentially the CPG serves switching off the motor neurons, which are driven during stepping by tonic depolarization, which comes centrally, and phasic excitatory input from sense organs. So they inspected the retractor, protractor, and levator motor neurons in the preparation which generates in the isolated mesothoracic ganglion alternating activity of the leg motor neuron pools. And here you see a retractor motor neuron recording in a rhythmic preparation. And when you measure the input resistance of the motor neuron during rhythmic activity, you see that there's a strong decrease in the interburst interval phase. And when you quantify, you see that that holds for all the recorded retractor motor neurons. And even nicer, when you play with the membrane potential in current clamp conditions, and this is shown here for a protractor and a levator trochanterous motor neuron, you can hyperpolarize them up to the point of the reversal of the inhibition without any phasic excitation building up, okay? And you can even reverse the inhibitory drive at interburst interval, giving rise to the conclusion that the CPG's pattern activity of the retractor, protractor, and levator trochanterous motor neurons, as we already know, by phasic synaptic inhibition. So that story would have been pretty boring, but then they started to record from the depressors. And then it became interesting, and it's still like that as we speak, because here again, you have the recording of the depressor motor neuron. When you look at the input resistance, there's not big of a change. And interburst interval, there's no drop in input resistance. And when you then change the membrane potential by current injector in current clamp conditions, the situation becomes exciting or interesting because you don't find a reversal of inhibition, but you get a buildup of phasic excitation. Which means that obviously the CPG controlling the trochanter of motor neurons drives the depressors by excitatory phasic synaptic drive, which gives presently rise to the conclusion that the organization of CPGs in the stick insect leg muscle control system cannot be regarded, or no longer, as a composition of modules with the same properties. So I've often said CPG and network and neurons. And so the question was about 10 years ago, what can we essentially learn in case we use animals which can be genetically approachable? For example, the fruit fly. So I did a sabbatical in Würzburg and in the lab of Martin Heisenberg, working together with Henrike Scholz there at the time. And after I returned, we started to work on the fruit fly in my lab as well in Cologne. There's a couple of questions that immediately come to mind when you know about animal walking, the scaling of animals, the neural control of walking, and that the initial questions would be, how does the fruit fly walking system compare to other walking systems, also larger insects? And two questions need to be answered, which is the control of interleg coordination, for example, when changing speed. And the question, is the fruit fly walking system organized in a modular fashion when it comes to neural control, as we've learned for the stick insect? So it was the graduate thesis work of Anne Wosnitzer, together with postdoc Till Bockemühl, where we started to analyze the footfall patterns of fruit flies walking at different speeds. Today, we do this completely automatically. This is unbelievable. At the time, you had to look at every single leg and essentially have to pinpoint the kinematics by hand. So what this slide shows is the footfall patterns of faster, slower, and very slow walking fruit flies. And you immediately see that these footfall patterns are quite reminiscent to what we know as coordination patterns in insect walking, the tripod coordination, the tetrapod coordination, and the wave gait. Tripod, always 3 legs are on the ground. Tetrapod, always 4 legs are on the ground, which are the open parts of the footfall patterns. And the wave gait, we have 1 leg on the ground at a given time. So obviously, Drosophila changes interleg coordination depending on the speed of locomotion. And there's one thing that I think is important to emphasize here. When we evaluate footfall patterns these days by high-speed video with automatic tracking software, what these systems often do is that they catch the state of the system at very brief time points, which means they simply look, where's the leg at any given time? Is it in the air or on the ground? And then they count the legs, and then it becomes clear whether it's 4 legs or 3 legs. But that's not interleg coordination. Interleg coordination is a functional and optical phenomenon which comes about as time goes by. So you cannot just count legs and make the probability plot in order to describe interleg coordination. So let's show this for the tripod. And we looked at this closer because the tripod looks like a nice, almost symmetrical gait. And we did so for the fruit fly. And we evaluated the quality of the tripod in a very easy way. We just asked the question, to which percentage are the legs that form a tripod doing the same thing at the same time? And divide this by the time between the first leg doing this and the last leg ending it. And what you can see nicely is that you have arbitrary values between 0.1, which is 10%, and 0.8, which is 80%. Which means here the legs are 10% of the time doing the same thing, and here 80%. When you look at fruit flies walking, you see that there's not a single state along the range of speeds of locomotion where they really walk tripod. They always are highly variable, and the quality of tripod changes as a function of speed, which means it becomes more and more variable as the animal becomes slower and slower. So tripod coordination strength in fruit fly walking changes as a function of speed of locomotion, indicating that also in fruit flies, there's no gaits. So the next question, is the leg muscle control system of the fruit fly organized in a modular fashion? And then we went back to a 70s approach, which some of you may know. It's removal of a distal leg segments of a leg, which is pretty simple because people did it before on larger insects like Ulrich Bessler, Gernot Wendler, and Fred Dalcombe. And here you see a slow walking fruit fly with the oscillating stump. You easily see that the stump has a faster oscillation frequency than the legs. Here you see a faster walking fruit fly, and you see that the stump obviously, well, it does a better job in coordinating with the other legs. For the time being, we evaluated the phase and so forth. I won't talk about it. For the time being, and for our question, one evaluation is important, looking at the cycle period. So in fast walking, the amputee, which is this one here, shows a frequency of oscillation pretty similar qualitatively to the front leg and the hind leg. In slow walking, it's much faster. And when you quantify data, this is the control animal, plotted the cycle period of a leg as a function of speed, you see that the middle leg amputee essentially does constant cycle period. The middle leg stump doesn't care about the cycle period of front legs and hind legs, as the front leg stump will do when you cut off the front leg. And even the hind leg stump only shows a minor dependence on speed of locomotion when it comes to oscillation frequency as compared to the other legs. So constant cycle period of leg stumps suggests segmental modularity of CPG networks for rhythmic locomotor activity in the fruit fly. The conclusion is that the fruit flies can serve very well as a comparable insect for studying walking because it controls interleg coordination comparable to many other insects. And the neural control of fruit fly walking shows segmental modularity. So then we turn to the questions, which neurons in the VNC are involved in generating leg stepping of the fruit fly? And here we teamed up with Barry Dixon and Salil Bidaye. And the challenge to study walking in the fruit fly is that you have to stop the fruit fly. This is now possible as Salil has identified descending neurons that can do the job and that can initiate forward walking. At the time, a finding was very helpful because fruit flies have the habit to walk forward. So we needed an experimental paradigm which allows for studying leg stepping in a controllable fashion, and that is walking backwards. The neuron that Salil had identified in his thesis in Barry Dixon's labs is the moonwalker descending neuron, which is here tagged with CS crimson, and when the red light goes on, then MDN is activated. And you see immediately, as long as the red light is on, the Drosophila will slowly but steadily walk backwards. So that was an ideal case. So we had a neuron that would generate stepping, induce stepping in a controllable fashion. So what we did then was that Kai Feng, Ryo Minigeshi, and Rai and Barry used the Transtango method in order to find postsynaptic partners of MDN. And even though that you may not see the details, this is simply to show you that they found many neurons in the VNC that essentially are connected to this neuron MDN coming from the brain. So what Kai Feng then did was he performed calcium imaging. So he coupled a calcium dye to those neurons in order to look at the influence of MDN on the postsynaptic partners. And together with behavioral assays, he decided, or we decided as we were discussing the data, to check out what this neuron MF09 would have as a function potentially during backward walking. Here you see that the descending axon from the brain of MDN comes in very close vicinity to the homolog neurons of type MF09, which was later on called LEN129. And here you see on the right-hand side a pair of those neurons in the metathoracic neuromere of the VNC of the fruit fly. So when you couple this neuron again to crimson and activate it with laser in a standing fruit fly, by the way, if you want to work on standing fruit flies, the best thing is to cut off the head. You see that each single stimulus exerts a leg lifting. Now we combine activation of MDN tagged with crimson and having tagged LEN128 with GTSAR2 now inactivated with blue light. You see that when blue light turns on, the hind leg is obviously not, or very rarely, finishing stance and switching to swing. And this has shown the kinematic data of the legs. And you see here the activation of MDN. The animal starts to walk with the legs nicely backwards. When you add up the inactivation of 128, stance phase goes on, and only very rarely you see the transition to leg swing. So silencing of this neuron leads to abolishing and delayed stance to swing transition in backward step. As we found one neuron serving this part of the stepping cycle, we also found another neuron that serves exclusively stance phase generation, which tells us that we can identify individual local interneurons serving the generation of specific phases of the leg stepping cycle for backward stepping of the fruit fly. A brief excursion to biomechanics. Because as you've seen, when you have the six-legged animal walking, it may sometimes slips the attention of the, two things slip the attention of the researcher. Number one is the legs are not alike. Number two, they operate in different operating ranges. And importantly, specifically here in the stick insect visible, you do have very slow stance phases and fairly quick swing phases. Which leads us to the question, what's the role of the antagonist in fast movements? Because as it's self-understood and known for long, passive forces are exerted by muscles which are not active. And in particular, one has to distinguish between the steady state versus the transient. What do I mean by this? Well, when you lengthen the flexor muscle, and this is the flexor muscle of the stick insect, very slowly, then you would get this green curve. Okay, if you do it like this in this experiment, in a time, you see that when you do it faster, you get much stronger development of force. And that's important because what we could show is that the muscle force gets much higher. The transient muscle force, resisting force of a muscle upon change of its length can be by the orders of 50 higher in the dynamic part of the stimulus as compared to the rest. And this is shown here. This is the steady state rest passive force of the flexor muscle. And this is the force generated by the inactive muscle when it comes to faster movements. And 800 degrees is a movement of the tibia that you can find at the beginning of the swing phase, easy. Together with other experiments, we were able to show that transient passive forces of antagonists can influence velocity profile during fast leg movements, to the fact that the decay of velocity during leg swing is highly likely to be under control of the passive resting tension, which is the transient resting tension of the flexor muscle. And this beautiful work has been done by Scott Hooper and Christoph Guschelbauer and Arndt von Twick. Finally, I would like to take you to a recent project that actually bridges my qualification, because we come back to posture control in the end. But now we would like to talk about multimodal processing in posture control. And this is work that Corinna Geberhardt did as a graduate student in my lab, initially together with Jochen Schmidt. So, as you've heard all along during my presentation, we have tons of load and movement sensors on the legs, some of which we have investigated quite well with respect to their function in generating the stepping pattern of motor activity, for motor activity. And these are the campaniform sensilla and, for example, the femoral chordotonal organ. So they induce specific influences on motor activity. For example, the tibial campaniform sensilla and the chordotonal organ have a specific influence, what one can call a reflex-like influence, on the extensor motor neurons moving the tibia. When you stimulate the femoral chordotonal organ, you get a nice resistance reflex opposing the movement of the tibia, which means this is a flexion, you get the extensor active, and the extension, you get the extensor inactivated. And you can do the same thing with the sinusoidal stimulus. Load feedback from the tibial campaniform sensilla, mimicking the stimulation of a subgroup, causes inhibition of the extensor. Okay, and here up on the in the upper traces, you see recordings from specific individual sensory neurons of the populations of sensory neurons in the respective sense organs. Like here, sensory neuron of the femoral chordotonal organ, or here as an example, a sensory neuron from the tibial campaniform sensilla. So the question was, and as some of you may recall, I did some of my thesis work on looking into pathways processing movement feedback to the extensors. So we want Corinna actually starting out with a slightly different topic. She ended up in looking closer at the question, how are the two proprioceptive feedback signals processed in the premotor network? So what she did was she stimulated movement and load sensors, specifically the chordotonal organ and the campaniform sensilla on the tibia and the trochanter, checked out the pathways to the motor neurons, and focused to some extent on the premotor network, from what we already knew, that for the chordotonal organ has distributed organization with supporting and opposing pathways ending up on the motor neurons driving the extensor muscle. So she recorded many of the neurons, most of which were identified, and she always found the same thing. This is shown here by a local premotor non-spiking interneuron. It's called I5. I because it inhibits the extensor. Here you see the current injection and the extensor stops to be spontaneously active. And when she stimulates the chordotonal organ, she sees a hyperpolarization with an elongation and a depolarization with a relaxation. And then we were looking for what do the other sense organs that she was checking do to the neuron. Well, the neuron receives inputs from all sensory neurons tested, which means from both modalities, movement feedback and load feedback. And from the load feedback, there's even 3 different kinds of load sense organs, the ones on the tibia campaniform sensilla, the trochanter, and the femur. So interneuron I5 processes input from all 3 sensory structures on the leg. Okay, is this an exception? We will look at this later, but first we look at the influence that such a change in membrane potential might have on the extensor. As we know, it's an inhibitory neuron. So this one will support will support the reflex activation, which means that the inhibition of this I5 will support the activation of the extensor. The opposite is true for the tibial campaniform sensilla when you stimulate the 6Bs as compared to the 6As, where it's supporting again, and so forth. So there's all when you look at the specific sensory influence that one sense organ has on this interneuron, the role for the motor output is either supporting or opposing. And the question is, was that an exception? No, it was not. Because there was not a single neuron that did receive only input from one sense organ, and even more, there was not a single neuron that did the right job all the time, which means that essentially the premotor network consisting of premotor non-spiking interneurons is operating for multimodal processing in a distributed fashion, with neurons supporting the extensor tibia activity or opposing, but doing it always in an individual mixture. So the question is, what do we make out of this? Because it simply makes the story a bit more complicated. And essentially, at the beginning, Corinna and me were hoping that adding load or looking into load would make the story easier, but it didn't. So we went to the next question and asked, well, potentially the situation becomes more clear when we look into the question how the two modalities influence each other. So Corinna stimulated now both sense organs individually and together. Here you see the stimulation of the chordotonal organ and the resistance reflex and the fast extensor and the slow extensor TBA. When you add load, you see, well, it's a bit less active. Yes, that's true. When you quantify the orange trace, and this is peristimulus time histogram, is below the pure movement-induced activity. The question is, does this change matter on the output side? In order to check for that, Corinna measured the muscle force generated under different conditions. And to cut this data set short, the black lines are the lines of extensor activation when you stimulate with different amplitudes, 20 degrees, 40 degrees, 60 degrees, the chordotonal organ, which is movement feedback. And when you add load feedback, you have the blue lines, which means force is decreased. And that means that load signals reduce the response amplitude to movement signals. Is that a change of gain or not? Well, you can say yes, but we just tested one value. So what Corinna then did was she looked at the spike frequency induced by stimulation as a function of amplitude. And she found that when movement signals were present alone, this is the dependence. However, when load was added, the values were not only smaller, the slope of the dependency was significantly shallower, which means that load signals reduce the response to and the gain of movement signals. The question is, how is this mediated? And in order to look at this, Corinna turned back to the premotor non-spiking interneurons and confronted them with the same stimulus regime, stimulating the chordotonal organ or the campaniform sensilla alone, and then adding both stimuli, which means going from unimodal stimulation to bimodal stimulation. If you would just add those inputs, almost arithmetically or linearly, you would find this gray line. However, she found the blue line, which is definitely smaller than the expected outcome. And this is shown here for this interneuron I1. You see the black trace for the movement stimulation, and you see the bluish trace for the movement combined with load stimulation, which has a shallower slope plus smaller values. 0 is here because this neuron is inhibited. She found as well a change for other supporting neurons. For example, E2, here she found a slight increase. And she went through all the neurons and the kinds of neurons like supporting and opposing, and always found specific changes or none. And that's important, because when you summarize the changes that she found, and I leave now just the pluses and the minuses to show opposing and supporting neurons as a function of gain of membrane potential. This is one, which means no change for some of the neurons. She found in supporting neurons, in some of them, a drastic decrease in the amplitude of response. Only for opposing neurons, she found for one, a drastic increase, which means that load chain signals change the gain of movement signals of pathways. Opposing pathways show an increase exclusively. Only supporting pathways show a decrease, and some doesn't show any change. So obviously, there's a highly specific integration of movement and load feedback within the individual pathways that leads to changes in the gain of their processing. And then this immediately, what comes to mind is a level of processing which has drawn a lot of attention in the past 20 years, which is the presynaptic communication between afferents. So the question that Corinna posed was, is presynaptic inhibition potentially involved in the influence of load on movement feedback? So she recorded from afferents from the chordotonal organ as well as from the campaniform sensilla and stimulated both sense organs. Here you see a recording from the chordotonal organ afferent. Here you see the stimulation with the movement. This is a movement sensitive afferent because with movement of the chordotonal organ, you see an increase in action potential frequency. You also see that there's in the background a depolarization coming in, and this actually has been described a long time ago by Boris and Matheson, and later on analyzed as well for the stick insect by Wolfgang Stein and Arne Sauer and myself. Essentially, this depolarization leads to a decrease in amplitude of the action potentials as indication and very direct indication for presynaptic inhibition. Okay, that's the known part. The interesting new part is that she often found that campaniform sensilla stimulation generates a drastic depolarization in the afferent, which leads to a systematic decrease in the action potential amplitude. So how can we test whether presynaptic inhibition of campaniform sensilla onto FCO neurons plays a role in this influence of load on movement feedback gain? Well, as you know, we found that there's a specific dependency of spike frequency in the extensors on stimulus amplitude without load and with load. Removing presynaptic inhibition is possible in the stick insect, as in other insects, by using a pharmacon called piperotoxin. When you do so, there's no difference in the slopes anymore, which means that the influence of load on movement feedback processing has vanished. Variability is high, but the quantification clearly shows there's no longer a change in gain induced by load feedback. So presynaptic efferent inhibition between load and movement signaling efferents contributes to reduction of movement feedback gain in the presence of load. What have I told you tonight? First, we talked about generating a task-specific motor output for leg stepping. The consequence is that the diversity of modifications in sensory influences and local processing falsifies simple concepts of descending control for the generation of motor flexibility. What I really have trouble with is seeing those green arrows leaving a brain in some textbooks with the word behavior written aside of it. Well, behavior doesn't come along, and particularly not motor flexibility, with a single arrow. What we've heard also concerns towards the systems level, the analysis on biomechanics. As we know, for our system and also for others, most likely, in small limbs, passive forces can contribute to the generation and control of velocity profiles of movements, which is actually a fact which is often overlooked by people like me that are mostly concerned with neurobiology and not with biomechanics and muscle properties. And finally, multimodal processing in posture control. In a distributed premotor network, gain changes can be mediated by relative weighting of antagonistic pathways, suggesting a mechanism for integrating multiple sensory modalities in a context-dependent manner. And that would not at all have been possible without those cool and great and nice people that I had the chance to work with currently and previously in my lab. Here's a list of names that I don't read to you right now. I have the fortune, I'm so fortunate to cooperate with colleagues and labs like Scott Hooper, Sasha Zill, Barry Dixon, Salil Bidaya, and importantly also Abdel El Manira. And we have profited immensely from collaborating with modelers and mathematicians and computational neuroscientists like Sylvia Down, Phil Holmes, Roger Quinn, and Nick Cichinski. And having said that, I'm done. Thank you very much for your attention.
Ansgar, that was fascinating. Can I start with a question?
Yeah.
Okay. Look, I remember that Bessler never believed in CPGs, and we had this discussion all the time. But looking at your results, I'm starting to think maybe Bessler had kind of a point. If basically you're showing that every connection has different mechanisms. You show that the inhibition, one is dependent on inhibition, one is dependent on excitation, but these are really, really small modules within your network. So what does the concept of CPG really helps us in this case? Because it's really like composed of millions of CPGs, or is it really like just a few connections that then get assembled in each step? I don't know. I mean, just
you can
probably complex.
I think this question, I don't want to be blunt, but this question, for the stick insect stepping control actually doesn't matter because it doesn't use the CPG as a clock-like function. It uses the network in order to drive the motor output in different states. There has been long discussions on oscillators, on state machines, and so forth. What we know as of today is that we can activate networks in the central nervous system of the stick insect that will generate independently rhythmic activity of the motor pools of the different leg joints, full stop. We can also show that the same neurons are active during stepping. I haven't shown that tonight because that's actually a long time ago that we showed this. But we also show that sensory feedback has access to individual of those networks and can change the phase of activity. And then we are back actually in some respect to the flight system, because the flight system, when you look at the flight system closely, and we did this a while ago when I wrote this review. There's 4 elements that can be cyclic active in the locus flight CPG. Okay, so you cannot just say this CPG is just one, you can sort of subdivide it. And the operation of the system when the stick insect is walking is definitely coordinated by sensory feedback, but I think it does not, I wouldn't overdo it in sort of, for me, essentially the knowledge that we do have CPGs for each leg joint helps to stable the activity, but not more. You are shaking your head.
No, it's fascinating.
No, yeah,
I'm like, I was so fascinated that, you know, like even the cycle period can be different left and right, and that's really like only a few connections that determine. So it's not like one CPG or CPGs. It's assembled each step in a different way. Anyway, and then you have your multimodal input, which is also highly distributed. It's
but that was only in standing. We have to be careful here. This is we have no clue what happens as soon as the animal essentially activates one motor neuron pool to do an active leg movement. Yeah.
Dirk, should I tell people, Dirk? Okay, go ahead.
Ansgar, the premotor non-spiker is these responses to the different campaniform and cototonal efferents all had, it looked like it, at least in the original recording, pretty distinct temporal dynamics in terms of sensitivity to velocity and acceleration and position and so on, which sort of suggests it's not just multimodal, right? So if you mix all of these things up, that you could get distinctly different responses at different walking speeds from it.
This is such a cool question, because yes, Dirk. What you see is when you look at the dynamic of the responses, and I think Corinna is here, and she may answer that because it's her work that I'm talking about. We have not played with the portfolio of sensory parameters, as you say, velocity or acceleration or position, in a combined or selective fashion. Okay, so that may have an impact. So I have no answer to your question.
Yeah, I'm very disappointed.
But there's one thing that I did not tell you that Corinna has found, and we have to finish this, is that these pathways, these non-spiking pathways, are not sort of non-collaborating identities, because they are horizontally connected with each other. And that hasn't, we knew from Malcolm's work that there some non-spiking interneurons have inhibitory connections. But what Corinna found is that, I don't know, what's the percentage, Corinna, of neurons that talk to each other? It was
almost all of them. Sorry,
almost all of them, yeah.
Portugal is killing my voice. Almost all of them are connected with each other. So there's super high degree of connectivity. And what Ansgar was referring to earlier, there's also the load signals are delayed relative to movement signals. So that might go a bit into the temporal dynamics of it. So if it's conserved in walking, then load signals will come in later than the movement signals of the same type of movement or same stimulus, which will probably also have all kinds of effects on the processing.
Joe.
Ansgar, one thing that really quite impressed me about your talk is the absence of mention of things like command neurons. And in crustaceans, command neurons are everywhere. They were first discovered in the swimmerhead system. They're in the walking system. They control everything. And using command neuron-based models, it was very easy for me to build robots with networks of electronic neurons that could control walking in any direction and show responses to feedback, etc. Why do you think insects are so different?
No, I don't get me wrong. not talking about command neurons doesn't mean that they are not there. The thing is that we simply have not done a lot of work on them. Actually, there's one paper recently published in Neuron that we worked together with Samlil Bidaye, where we showed command neurons for forward walking. And this neuron MDN, the backward walking neuron in fruit fly, that's a dynamite command neuron, okay? So yes, we do have, in the meantime, we also have command neurons for forward walking. There's one which is a descending neuron, P9. It had been known from earlier work in the fruit fly to get input from visual system. And it's bilateral, and essentially you can also induce curve walking by activating either or. But when you activate both, you can essentially start the machine to walk forward, straight forward, okay? So yes, Joe, you can account it to the incompetence of the researcher that he could not in his work do research on command neurons in the stick insect. Does that answer your question?
I was a little intrigued by, did you say that there are no gates? You know, like there's no, basically there's no gate, and
no,
and that's crazy.
No. The term gate in insects is for the studies on is for most of the studies is a pure invented term by the examiner. There is, because you see, when you look at the old work on Alexander on quadruped walking, quadrupeds have gaits, and there's one thing which tells you about the existence of gaits. You cannot, when you go through the speed in walking of a quadruped from walk to trot to pace to gallop, you cannot get away without one leg doing an intermittent step in order to fix the phase difference at one point. And actually this, to be honest, this drives me nuts when people talk about gaits in insects, because I call them coordination patterns, because you can do all the coordinations we know in the insect, you can create by having a front to back sequence of leg lifting, swing phases, and by just making the stance phases shorter and shorter, okay? So there is a few exceptions, and that is cool. And there's Tom Wyman. He's a junior. No, he's not a junior anymore, but he's an independent researcher in my group. And when you have very small aphids, for example, very small 6, 8, 10-legged and other invertebrate species, when they speed up and when they become faster and faster, the dogma is that the faster it goes, the more it becomes tripod. For those small ones, the tripod falls apart again and they switch back to wave gait because it's a more stable situation. It's really stunning. So, but gait and insects in general don't go together well.
Karen has
Karen.
a question.
So I have a question.
Karen, hi.
Yeah, so I want to go back to Nino's comment about the functional architecture of central pattern generators. So tell me, Ansgar, if you believe this or not, or if you agree, but it seems to me that locomotor patterns that are more stereotypical, the ones that are faster, less flexible, you think they're more reliant on CPGs for their coordination? And so the slower the rhythm, especially in stick insects, and the more flexibility there is, the more adaptation is needed, then there's a shift, a continuum to the integration of more sensory input. And so the CPG functional architecture is very fluid, and I see that it shifts to more reliance on the CPG, which is very much needed when we have this less flexible, faster rhythm.
There's, Karen, I cannot agree more. And there's one piece of evidence from the fruit fly project on the stump oscillation, because I just referred to cycle period. When you do the phase evaluation, this is, and then what you see is it's a weakly coupled oscillatory system. What do I mean by that? With slow walking, you have the 10 cycles per second oscillation of the stump, almost phase free to the other legs. the faster it goes. It's a bit like at 12 to 13 body lengths per second, the variability goes down, and then it appears almost to lock in at 0.5. I cannot say whether this is CPG or not, because this is an almost intact animal with all the sensory feedback on the other legs. But as fast as they walk, and they have cycle periods of let's say 15 steps or 16 steps per second, which makes it hard to believe that sensory feedback can be acute and concrete enough to do a big job. I would not be the least surprised to see that being put in charge, but then it would be a coupling strength dependency across cycle period range needed for locomotion.
Yeah. Okay.
I don't know, was Dirk first or Joe first?
Joe.
Joe.
Two questions. One, Ansgar, how's Gernot doing?
Gernot he's doing fine. Gernot's wife died about 8-9 years ago, and Gernot, but he's living in close to Bonn now, and he's doing fine. He doesn't come to the lab, and he is doing a lot of hobbies. That's what I know.
And my second question is, do you think there is any one synergy within the leg pattern generator in the insect that serves as a neuronal oscillator? In other words, the site where perturbation inputs hit and is modulated parametrically to control the speed of walking.
That's a good question. Actually, sometimes there is an early study by Volker Dürr and also work from Hol Kruse, where they hypothesize that when you talk about rhythmic leg movements in insects, here now I'm only talking about the stick insect, okay? Then essentially what walking may be is a specialization of searching movements, where a specific phase of the movement is arrested by load information and ground contact, which means by pure mechanical input to the system. Because for the searching movements, you do have essentially a very regular ongoing cyclic activity. Yeah. So that could be, but but that's the only idea I have.
I guess maybe if I could rephrase it, if there are interleg coordinating units, they have to impinge on some target that they perturb. And where do you think that target lies? For example, in the swimmerate system, there's a coordinating interneuron within each segment that receives ascending and descending inputs from other segments. Do you envision anything like that in the stick insect?
Actually, there's yes, and the argument goes as follows. When we showed the independent oscillators for the modular pool of each leg joint, we found that every once in a while, the rhythms of the three main leg joints team up in one pattern, and then it falls apart again, and then it comes back. And so we recorded, but we never went more systematic at this. I recorded from interneurons, from local interneurons that essentially can induce coordinated activity between, and this is in the 95 paper where I recorded from premotor interneurons during rhythmic activity in the deafferented nerve cord, where I send you the ref, and then you can check out whether this is what you mean. But yes, I would agree.
Sort of piggybacking on what Joe said and also what Karen said, does this, I mean, is this even clear that there is such a thing as insect walking, that term makes sense in the sense that on the scale of a slow walking stick insect and a I don't know, cockroach running at top speed, the sort of the importance of CPGs and central coordination and the way sensory feedback is integrated and is important. Is it a continuum, or is it really sort of more like, let's say, insect flight, where, you know, the way between synchronous and asynchronous flyers across different species, it's sort of like, well, sure, there's similarities, but they're fundamentally different concepts behind the control mechanisms there.
Well, there's.
I mean, it's hand waving, but sure. But
Yeah, no, but my answer will be similarly waving my hand instead of yours. But let's put it this way: for the, you might remember when I said that when I talked about interleg coordination. Now I'm not talking about one leg, but the coupling between the 6 legs. Okay, and I think the same holds for controlling one leg. Is that essentially, whether it's in a continuum, I cannot account on, but I would definitely think that the internal mechanisms of network control are changing gains or influence impact or strength between sub-networks or components in a way that essentially you could create such a continuum. You see what I mean? Because the cockroach—
are you talking within a species or across species? Or
within a species, because, and this is one thing which often happens to us, is that we think, oh, the cockroach is fast and the fruit fly is fast and the stick insect is slow. This is true and wrong in the same way, because we should not mix up the fact that the fruit fly often walks fast and the cockroach mostly walks fast with the fact that they can all walk very slow. And as soon as they walk slow, you find the stick insect. Because that actually was the story starting when I read this paper by Heisenberg and Wolf about Drosophila walking from 1990. And when they said, oh yeah, Drosophila is a tripod walker and it's all centrally coupled and full stop, it's this J. Confiseol paper. And then I said, wait a minute, you don't have data on the lower end of the speed, you just have data on the upper end. And so essentially, Till Bockemühl was not pretty amazed when I asked him to look for slow walking fruit flies. He said, oh, come on, they don't like to walk slow. I said, we don't study emotions of fruit flies, we study walking. So please look at slow walking fruit flies. And it took him quite a while and he found them. And when he evaluated them, he found wavegate. And this is what drives, and sorry to say, this is what agitates me, obviously, at the moment as well. I can control the blood pressure, but which agitates me, that we essentially mix up the description of the behavioral need of the behavioral ecological need of an animal with the neural organization. No, that's wrong. Okay, and so actually the wildest hypothesis on sensory central, Nino may recall it, had Keir when I was a postdoc with him, and this was made Keir Pearson at the time. It was never published, but we had a guest researcher who did a model. Nino, you recall this depressor elevator model, because Keir had the idea that when the locust would start flying, it would start flying with a CPG weighted motor activity, and when it would then go on long-term flight, then it would become more and more like almost automatic, dependent on sensory feedback in order to drive the system to keep it going. Okay, so no answer, but stories.
I have one last question.
Yeah.
Now, the stick insect is really quite a specialized animal.
Yeah.
Do you think that neurophysiology would be better served by choosing a more generalist animal?
Yeah. Yes, sir. Yes, sir. I've often thought about it. And
that's why I said swimming in bricks.
Guys, you really want to increase his blood pressure.
No, I love this. I love this. Look, I actually in the mid-90s, I started to work on the locust together with Harold Wolf, and then I did some additional work in Cologne when I was here. There's a couple of things with the locust, which it has a dynamite topology knowledge, because the lab of Malcolm Burrows has characterized so many neurons and connections. But the problem is that when you look at the locust walking, it's interesting, but I would not quote it as walking, because in many cases, you can make a locust walk on a treadmill if you keep it up, and then it will not drag its body along the surface, which is a problem for me to consider it as walking You see what I mean, okay? And then we do have the hind legs. Anyway, so yes, I've looked around and I have not yet found an animal except for the fruit fly, which offers simply an additional set of tools that we don't have on the stick insect that I would like to work on. But I'm only 60, so there's some time left for looking around. So I'm no problem. No, yeah. I'm
No, when I
reconsidering.
started my thesis, I worked actually on initiation of locomotion.
Yeah.
And it just drove me totally nuts because I didn't know, is this like a movement, or is this like a step, or is this like just a twitch, or like, it wasn't really the clear onset of a behavior. It was great. That's why I switched to flight, because there you know, you can either fly or you crash, but with locomotion, you have all these in-betweens, which is really a hard project. And I think, Ansgar, your talk was like clearly confirming this. This is incredibly flexible, incredible multimodal, and tons of little units. It's daunting. Yeah. Really tough brick to solve.
A tough brick to make swim, or what?
Yeah.
So I would like to comment on that notion that we should choose an animal model system that is a generalist, that is exhibiting a generalizable behavior. I'm not sure that that's the best way to go about things. I think traditionally the way to choose model organisms has been to pick someone who is an expert at something, who is a specialist at does something is extremely well, like the barn owl with the auditory localization, right? So it's kind of weird for me as a computational theorist kind of person to say this, but I think that the choice of model organism should not necessarily be a generalist, but should be a specialist.
Well, can I comment on this, Astrid?
Yeah, absolutely. Yeah.
Sometimes I have the impression that, yeah, I would say you're right, but from a second point of view, I would say perhaps sometimes our valuing as some animal or some organism being a generalist comes from the fact that we don't know its ecological needs and behavior sufficiently well, and then essentially I value comparative research, but that for 20 years in Germany was almost like, if you would say you would do that, you would be immediately old-fashioned. But that came from a development that was not very good, because it became to a self-fulfilling argument, which means if you want to get rid of criticism why you do interesting but not relevant work or whatever, then you would say, I'm doing comparative work, which means that don't ask me because you cannot understand what I'm looking for, because I'm doing comparative work. And I think we have to reconsider this notion, because I'm not so sure how many generalists are around.
I would go a little further than Astrid too. I would also make the point that after your argument, you should work on a model system or a model animal that is particularly good at something. And I'm not sure that the stick insect qualifies as a particularly good walker, but this is just to piss off Ansgar again. But no, but yeah. It's sort of like the comparative approach means you only, if you want to understand general principles, generalizable principles of neural control of locomotion, you can only know that if people study different things. I mean, otherwise you never know what's generalizable, right? So there is no argument to be made to find a particular type of insect, for example, unless you're interested in coming with the best or broadest applicable question to the answer of how insect walks. But that's not your, I mean, I'm assuming that's not your main motivation of doing any of this. You want to find generalizable control principles for locomotion in general, right? So from that argument, I mean, the insect, the stick insect is a great system to do this on. And it's and then you kind of hope that someone else does this on the cockroach and someone else does it on the locust, but you don't have to do everything.
Mhm.